Today we are going to discuss about immunoglobulin E, otherwise called the IgE. So in detail of that molecule that we will look. So this antibody is basically made up of the epsilon heavy chain while kappa or lambda light chain. So because of this epsilon heavy chain it is called IgE. Same as a immunoglobulin G that is made up of the gamma heavy chain while kappa or lambda that is light chain. Further in detail of IgE, it is also called a 8s antibody because sedimentation coefficient that is 8s while molecular weight that is 1,90,000 to the 2 lakh Dalton. Further in detail of this IgE that we will look, so it is also called a cytophilic antibodies. They mediate allergy, hypersensitivity and anaphylactic reaction. When certain antigens such as penicillin are injected a few times, here you have to remember few times. So mostly that first time any antigen that is not creating such kind of condition like hypersensitivity, but when injected for the many times, so mostly from the second times onwards, that type of reaction mostly seen. So IgE class antibody are produced which anchor on mast cell. Basically this IgE that is present over the mast cell. So when the same chemical is injected next time, means second times or any time further, then antigens fix on this such antibody. So whatever the molecule which is working as an antigen or foreign substance that will fix over the antibody, causing mast cell degranulation and release of the histamine and slow releasing substance, or short form that is called SRS. So in detail exactly how it is occurring that we will look. First you have to remember IgE antibody or this immunoglobulin have the property to fix over the mast cell and basophils. So they are present over this mast cell. So here in the picture it is shown over this mast cell that particular antibody IgE is attached, and that attachment with the help of this Fc receptor. So with the help of this Fc portion of this antibody it is attached over the mast cell, while FAB that is fraction antibody that is helping to attach over the antigen. So once it is attached over this mass particular antibody immunoglobulin IgE, whatever the granules that is present inside the mast cell that will be released into the surrounding. So that is called degranulation and release of the granules content into the surrounding tissue. So that granules mostly containing histamine and other substance that mediate allergic reaction, or called a slow reacting substance. So what is the effect of this molecule that we will look. So this reaction that can lead to the vasodilatation, means broadening or dilation of the particular vessels, hypotension due to that dilatation of the vessels, hypotension occur, as well as sometimes bronchial constriction is seen. This is the basis of penicillin anaphylaxis, as well as hay fever caused by the fungus, asthma by the pollen, and urticaria by the absorbed food elements. So all this hypersensitivity that is based on this reaction or release of this particular histamine from the mast cell. Further, the peak of this reaction will be at about 30 minutes, hence called as the immediate type of a hypersensitivity. IgE level in serum is markedly increased in a helminthic infection also. But most important thing that you have to remember, it is released in a allergic reaction but does not fix as a complement. So you have to remember this complement fixation that is mostly occurring with the help of IgG or also with the IgM, but not with the IgE. So that's all are the important thing about the immunoglobulin E.